I'm Brent.
I'm Brett.
I'm Emily. And I'm Maddie.
And we're here to talk
about cellular respiration.
To begin, the equation for cellular respiration is C6H12O6 plus 6O2 gives you 6 CO2 plus 6 H2O plus ATP, or energy.
The first major step of cellular respiration is known as glycolysis, which occurs in the cytoplasm of the cell. It begins with glucose, which is a stable molecule. Glucose must be made a little bit unstable so that it can be broken apart into smaller molecules, which will release energy. Energy will be used in two ways. Some will be used to make ATP, and some will be transferred into the form of hydrogen and electrons to the electron carriers. These electron carriers will carry this energy to the electron transport chain. To make glucose unstable, phosphates are added to the glucose molecule. These phosphates come from ATP, so in glycolysis, 2 ATP molecules are spent right away. The glucose molecules now become 2 molecules of 3 carbons and a phosphate attached. These molecules are called G3P. The G3P molecules will then have another phosphate added to the cytoplasm. The G3P also becomes oxidized, losing electrons by an enzyme. NAD plus will pick up 2 electrons and a hydrogen from G3P. NAD plus becomes reduced to NADH. The molecule now has a lot of potential energy. It is a 3-carbon molecule with 2 phosphates now attached. ADP from the cytoplasm will now take a phosphate from this molecule and make ATP. When ADP takes a phosphate from a molecule to become ATP, it is called substrate level phosphorylation. This occurs a total of 2 times for 2 molecules of G3P, so 4 molecules of ATP are made. But since 2 input molecules of ATP were used in the beginning of the reaction, the net ATP is only 2. The two 3-carbon molecules are now called pyruvates. The final products of glycolysis are 2 NADH molecules, 2 molecules of ATP, and 2 molecules of pyruvate.
This brings us to the next step, which is pyruvate grooming, and it occurs in the mitochondria. We start out with 2 molecules of pyruvate. 2 CO2s are released as a waste product. And then 2 NAD pluses are reduced by having electrons and a hydrogen added to them to make 2 NADH. Coenzyme A is then added to this, and we have acetyl-CoA. The final products are 2 CO2, 2 NADH, and 2 acetyl-CoA.
This brings us to the second step after pyruvate grooming. This major step is known as the Krebs cycle, or citric acid cycle, or tricarboxylic acid, or TCA.
The acetyl-CoA that leaves pyruvate grooming loses its CoA, or coenzyme A, which then returns to pyruvate grooming. This leaves a 2-carbon compound, or molecule. When this enters, it immediately combines with a 4-carbon compound known as oxaloacetate. This gives you a 6-carbon compound known as citrate. Following this, the citrate is oxidized, which means it loses electrons, and it also loses a carbon, which leaves the cycle in the form of carbon dioxide. Because citrate was oxidized, the NAD plus is reduced to become NADH. Following this, the 5-carbon molecule left is known as alpha-ketoglutarate. The alpha-ketoglutarate loses another carbon, which is also released in the form of carbon dioxide waste. Another NAD plus is reduced after alpha-ketoglutarate is oxidized, and the NAD plus becomes NADH. When this occurs, an ADP joins with a P, or a phosphate group, to form ATP, known as substrate level phosphorylation. This phosphate comes from the substrate. Following this, you are left with a 4-carbon compound known as succinate. The succinate is then oxidized, and another electron carrier known as FAD is reduced to become FADH2. Following this, you are left with a 4-carbon compound called malate. This is oxidized again, and NAD plus is reduced to become NADH. When this occurs, you are left with your final 4-carbon compound known as oxaloacetate, which then combines with the 2-carbon compound from acetyl-CoA, and the cycle begins again. The final products of one glucose molecule from the Krebs cycle are 6 NADHs, 4 carbon dioxides, 2 ATPs, and 2 FADH2s. From one acetyl-CoA molecule, you get 3 NADHs, 2 carbon dioxides, 1 ATP, and 1 FADH2.
The third major step in cellular respiration is the electron transport chain. This occurs in the cristae of the mitochondria. This step can happen because of all the electrons from NADH and FADH2 that were made in glycolysis and the citric acid cycle. High-energy electrons from NADH and FADH2 accumulate in the inner membrane of the mitochondria. Electrons enter the cristae and are passed from carrier to carrier. These carriers are proteins embedded in the membrane. One of the carriers is called cytochrome C. This protein is found in all eukaryotic organisms. The electrons move from carrier to carrier because of differences in electronegativities. The electrons will be passed through carriers in the membrane until it reaches the final electron acceptor. This is oxygen. The energy that is released as electrons move through carriers of different electronegativities is used to pump hydrogens across the membrane to maintain a concentration gradient. The hydrogens move down the concentration gradient through the protein ATP synthase to make ATP. Remember that this is called chemoosmosis. When ATPs are made, this way is called oxidative phosphorylation. The hydrogens are then bonded to the O2 to form water. We also breathe out water just like we breathe out CO2. If we do not have oxygen, NADH and FADH2 cannot dump off the electrons and hydrogen because the system will back up. If the hydrogens cannot flow through ATP synthase, no ATP will be made. If the system backs up, aerobic cellular respiration cannot continue. The final products of the electron transport chain are 34 ATP. 30 of the ATP come from the 10 NADH, and the other 4 come from the 2 FADH2.
All the steps so far have been a part of aerobic respiration, where they occur if oxygen is available. If oxygen is not available, anaerobic respiration occurs. This is also called fermentation. There are 2 types of fermentation. There is lactic acid fermentation, where the NADH gives its hydrogen electrons directly to the pyruvate, and this forms lactate or lactic acid. And this occurs in higher organisms such as humans. And our muscles do this when they are working hard for ATP and they can't get enough oxygen to them, so they have to produce ATP by fermentation. The byproduct of this is lactate. This is what causes muscle soreness. The other type of fermentation is alcohol fermentation. This occurs when the pyruvate from glycolysis loses a carbon in the form of carbon dioxide. The remaining molecule is a 2-carbon molecule known as acetaldehyde. The NADH can give acetaldehyde its electrons and hydrogen to become NAD+. Again, this will be important because NAD+ can now be used again in glycolysis. When acetaldehyde picks up the hydrogen and electrons, it becomes alcohol or ethanol. There are 2 types of anaerobes, strict anaerobes and facultative anaerobes. A strict anaerobe has to live in an oxygen-free environment because oxygen is toxic to it. A facultative anaerobe, they like aerobic environments, but they can survive in anaerobic environments. This is what yeast is. And then ethanol, yeast dies when alcohol content reaches 14%.
This concludes our cellular respiration lesson. We thank you for watching, and we hope to see you back here soon.